right, part 3. So, phospholipids. These are a member of the lipid family. Similar to that of triglycerols, they include glycerophospholipids and sphingomyelin. So they're variations of the triglyceride structure that we looked at. These, I guess the most obvious thing about phospholipids is that they will contain a phosphate. We'll see that that includes two classes, glycerophospholipids that also have a glycerol, and sphingomyelin, which contains sphingosine. So the goal here, draw phospholipids that have either glycerol or sphingosine in them. So let's take a look first at glycerophospholipids. So these contain two fatty acids, normal to the triglyceride fatty acids that we've seen before. First and second hydroxyl groups of the glycerol. Now, the hydroxyl group, the third one, forms an ester bond with phosphoric acid, and then that forms another, we'll call it a phosphoester bond, with an amino alcohol. So there's an ester bond here, and a phosphoester. There. I'll show you the structures of these in a minute. Just, for now, use these labels as just to sort of orient yourself as to what is included in these different structures. So glycerophospholipids have a glycerol, a phosphate, an amino alcohol. Sphingomyelin contains a sphingosine. So sphingosine is different than glycerol. Sphingosine also has a big long chain on it that kind of resembles the glycerol with one fatty acid on it. Right? Glycerol, if it had a fatty acid on it, kind of looks like sphingosine. So sphingomyelin contains a fatty acid, just one of them. Normal, actually this doesn't even, I don't even think this is a normal bond. We'll look at it here in a second. I think this is an amide bond as opposed to an ester bond. And it contains, again, a phosphate group. I think this is the ester, and then another phosphoester bond to an amino alcohol. Now, differences in sphingomyelin will be differences in the amino alcohol. So let's take a look at those. So amino alcohols, there's a few. These are found in glycerophospholipids. They're also found in the sphingolipids, or sphingosine. This is choline, this is serine, and this is ethanolamine. These are all ionized at the pH 7.4. This is important. We'll talk about this later too when we look at the amino acids and amines, because at physiological pH we often see these charges on the nitrogen compounds. But just realize here that this gives the polar part to our otherwise nonpolar lipid. So we're going to end up with a molecule that is partly polar, partly nonpolar, and the reason that it's polar is going to be because of not just this, but the phosphate as well. Phosphate has a lot of charges on it, and these amino alcohols, they are going to be the polar bits. So let's take a look at some of these glycerophospholipids. So we can take a glycerophospholipid and put choline on it, and we would have a category of phospholipid called lecithins. And we could also put a different amino alcohol, in this case ethanolamine, and that could give us the category cephalin. These are two types of glycerophospholipid. They're abundant in the brain and nerve tissue. We find them in egg yolk and wheat germ and yeast. They're going to be abundant in different parts of the body, depending on where we need them to do these different jobs. I forget how exactly they're named. Let me see if I can remember if it's going to come back to me. I don't believe so. I go over it in more depth in one of my other classes, but here just know that there are some categories depending on which amino alcohol you get. Now, glycerophospholipids do contain both the polar and non-polar regions. This allows them to interact with both polar and non-polar substances. So if you take a look at a glycerophospholipid here, you can kind of clump all the non-polar sides on the left here, or the right, and you can kind of put the polar parts on the right. So the negative charge there on that, the positive charge over here. Again, polar. We often sometimes use the terms head and tails when we talk about phospholipids. We say that they have a polar head and a non-polar tail. Sometimes we even just show these little representations here. I remember when I was in school, they were circles, but we do this. And these little things represent the hydrocarbon fatty acids, and then these little head groups represent the ethanolamine or the amino alcohol on the phosphate. And we sometimes draw these so that we can show a lipid bilayer. So if you know what a lipid bilayer is, it's when you have two layers, right, of lipids where all the nonpolar parts are interacting and all the polar parts are on opposite sides interacting with water. This is what our cells are made of. This is like the boundary that encapsulates a cell. Um, so sometimes they're represented this way, but they definitely are different in their polarities. All right. Draw the condensed structural formula for a cephalin that contains two stearic acids, a phosphate, a serine. Yeah, the serine would be the amino alcohol here. I don't think we talked about serine before. So cephalins can have serine and ethanolamine. The lecithin has the choline. So here you're drawing that other amino alcohol. So pause it, give it a shot. I think this actually walks you through it. So we're given everything. The bonds that you're interested in are the ester bond to the phosphate, the phosphoester bond to the serine. So where on the serine, remember the serine's an alcohol, so this side here would have had the alcohol part. All right, another learning check. Identify each of the following as either a fatty acid, a triglyceride, an alcohol, or a glycerophospholipid. So pause it, give it a shot. So hopefully you recognize choline as an amino alcohol, and glycerol trioleate as a triglyceride and not a fatty acid. Cephalins can only be phospholipids. So sphingomyelin contains sphingosine. Sphingomyelin has an amide bond instead of a ester bond. I think I'm going to show you that here in the structure. There's also a hydroxyl group that forms the ester bond with the phosphate. So we'll take a look at sphingomyelin here more in depth. Now, sphingomyelins, these are abundant in the brain, in the white matter, and in the myelin sheath. This is the coating around the nerve cells. I can't spell. Myelin sheath. This insulates the nerve cells so that it can send signals over a distance. When you have something like multiple sclerosis that attacks the myelin sheath and degrades it, it leads to nerve cells not being able to conduct signals, and then your body's unable to sort of do the things it wants to do. The scars on those neurons from the degradation of the myelin sheath impairs transmissions of nerve signals. So this molecule here is really important for us to being able to send electrical signals through our nerves. And so by coating those nerves, we actually increase the speed of those nerve impulses. So that's where sphingomyelin is most abundant. Now you can see here what we're talking about. In glycerol, in this position we had another OH, right? It was a triol. In sphingosine, there is an OH here, as well as one here. But there's actually an NH. This isn't even showing you sphingosine. This is showing you um, sphingomyelin, but sphingosine would have had no bond there, and it would have just had another H, NH2. But instead now we have what's called an amide bond. So there's an amide bond there joining the fatty acid. So that one, when you're drawing, is a little bit different because you tempted to put an O there like we do with glycerol. And then again, the hydroxyl here forms the phosphate group, and then our choline, this is where the other OH would have been. So sphingomyelin, sphingosine, myristic acid, and choline. But again, the important thing here is to know the difference between the sphingosine molecule and the glycerol molecule when you're identifying these. All right, another learning check. Draw the condensed structural formula for this sphingomyelin, one that contains palmitic acid along with the choline, amino acid choline. So give that one a shot. Should look something like that. Again, focusing on the fact that nitrogen usually wants 3 bonds unless it's got 4 bonds and a charge. So that's what it means with ionized amino alcohol. Don't forget the negative charge on the oxygen there in your phosphate. All right, last section, I think, steroids. So these are structurally very different. So the learning goal here is going to be able to draw the steroid nucleus and compare the structures and functions of various steroids. So what I'm showing you here is relating to the high and low density lipoproteins that transport cholesterol. We're going to look at some of those. This is what happens when you have cholesterol buildup. So this is cholesterol clogging an otherwise what should look like a normal blood vessel. When cholesterol sort of clogs it, obviously your blood can't flow, and this has to do with cholesterol either being deposited or not being able to be scavenged out of your blood, and we'll talk about which proteins, I guess, are responsible for that and which chemical messengers are responsible for that. So all of the steroids contain this nucleus. So you can see it's some cyclohexane rings, it's a cyclopentane ring. Again, pay attention to how they're fused together. Numbering, we're not going to be numbering or naming substituents, but it is worth paying attention to how this numbering scheme sort of goes. 2 methyl groups. There are 2 methyl groups at position 18 and 19. So those are methyls. And you'll see that a lot of variations on this backbone. One of the first ones we'll take a look at is cholesterol. So you can see right off the bat, cholesterol has some additional structures on the top of that last ring there, also a double bond and a hydroxyl group. This is one of the most important and abundant steroids in the body. So hydroxyl group on carbon 3, double bond between 5 and 6, methyl groups 13 and 10, and an alkyl chain at carbon 17. Cholesterol is obtained from meats, milk, eggs. We get it in our diet. Most of it, though, we actually synthesize for ourselves, but it does come in large part from our diet. It's synthesized from the liver, so the liver makes it. We need it for our cell membranes. We need it for brain and nerve tissue. We need it to be the starting material to make steroid hormones. And we also need it to make vitamin D. It'll clog our arteries, though, if the levels are high enough, and plaques can actually form. Cholesterol is considered elevated if you have more than 200 milligrams per deciliter. And even though it is synthesized from the liver, we can obtain it from food, and so having a diet that's low in foods containing cholesterol can be helpful in reducing the amount that's actually in your blood. If you take a look at this table, you can see different foods and the different sort of cholesterol content in those foods. Obviously, something like meat and eggs are going to have the largest sort of component of that. Quick learning check. So match the component of the cholesterol molecule with the following. So find the carbon chain, the steroid nucleus, the hydroxyl group, the methyl group. Shouldn't be too difficult here. Hopefully that was pretty straightforward. Again, we're just identifying the different parts of this steroid molecule. Now that was cholesterol. Let's look at something else. Bile salts. Bile salts are synthesized in the liver from cholesterol and stored in the gallbladder. So these are kind of like your body's soap. It uses this to help you digest fats. They have polar and nonpolar regions, and they literally do. They act like soap to help you absorb cholesterol and other fats from your diet. When a large amount of cholesterol accumulates in the gallbladder, though, we can get gallstones. That's what I'm showing you here. So these are gallstones just from a large amount of cholesterol having kind of piled up in there. This is the bile salt. So sodium glycolate is a bile salt. You can see the steroid nucleus here, the sort of modification here that makes it polar. So this comes, let's see which part of here. This cholic acid is where we're going to get the majority of this sodium glycolate. And then glycine, the amino acid, provides the rest of this sort of structure over here. Our body then mixes those together, and that's what we get. So cholesterol probably, when we add some amount of acid, carboxylic acid functionality, becomes cholic acid. So sodium glycolate is the bile salt. Again, nonpolar and polar regions acts like a salt. Now, lipoproteins are also important because a lot of these molecules are mostly nonpolar. They can be really hard to transport around our bodies. So we have these things called lipoproteins, and these are molecules that we combine with lipids to make them more soluble. We typically will take glycerophospholipids and proteins and mix them together, and they're called lipoproteins. One of the molecules that gets transported by these lipoproteins is actually cholesterol. Cholesterol is transported as this cholesterol ester. So you can see it's very, very non-polar here, which means it would not mix with our blood. And so we need lipoproteins that are both polar and non-polar in just the right ways, right? Non-polar enough that it likes the cholesterol, polar enough that it likes the water, and so these things can roll around in our bodies. And so you can kind of see here, the lipoproteins will surround the non-polar lipids with all of their non-polar parts. And if you zoom in here, you can see all these little non-polar tails. And then the polar heads are on the outside of the lipoprotein in contact with the water and blood environment, or the blood and water environment, I should say. And they're soluble because the surface consists of the polar lipids. It has all those phospholipids and the phosphate groups and the amino alcohols. So you can see that here, phospholipids. Now, cholesterol also is embedded within the walls of our cell. So these aren't cholesterols being transported like on the inside here. Cholesterol is a molecule that we can use to stabilize or to make our cell walls more rigid or less rigid. Sometimes warm temperatures mean that there's too much motion and too much fluidity, and so by sticking a lot more cholesterols in here, it helps keep everything together, helps pack everything together, make it more solid. Likewise, we can, if the temperature's too cold and we need more fluidity, we can take out, or maybe I said it the wrong way. We put in cholesterols to make the bilayer more fluid. We take them out and everything can pack together better, and that makes it more stable or more rigid. And so the varying amount of cholesterol can vary the flexibility of the outside of our cells. So that's one of the major roles that cholesterol plays in our membranes. But again, lipoproteins are designed to be able to transport non-polar things around our bodies. You can see here some of the different lipoproteins. They differ in their densities, their compositions, and their functions. We often name them or talk about them by their density differences, like very low density, and very high density, or just high density lipoproteins. Here, the density is just how much weight they have per size. So we have chylomicrons, we have very low density lipoproteins, low density lipoproteins, and high density. And you can see here what percentage of each lipid they carry. So chylomicrons, for example, carry mostly triglycerols, where very low density carries more cholesterol esters than the chylomicrons does. Low density carries the most amount of cholesterol, or cholesterol esters. High density only has some, but it carries the most amount of protein. Protein is what gives it that mass, which makes it so heavy. So because it has more protein in it, it's got a higher density. And you can see that kind of correlates with the amount of protein. As it goes down, the density goes down. Now, both of these lipoproteins, HDL and LDL, they transport nonpolar lipids and cholesterol to cells and into the liver. They take it from the liver to the muscles. The HDL, we call it good because it runs around sort of cleaning your body of cholesterol. Its job is to go and scoop up the cholesterol either from some of these other lipoproteins or just the free-floating cholesterol that's in your blood. LDL is the one that takes cholesterol that's just been synthesized and transports it to the cells. And so if the cells are already packed with cholesterol, LDL ends up dumping it in the arteries and just leaving it around. And so that's why it's considered bad, is because it's the one that carries cholesterol. But you can see here there are recommended amounts of cholesterol that we should have, and you can compare this to your diet, or you can see if you happen to have high levels of cholesterol, it could be something either that you're making too much of it, or more likely it could be just that you've got too much of it in your diet, too much of it going into your body and not enough of it being used. Let's talk about some steroid hormones. So these are chemical messengers. They are produced from cholesterol. We're going to look at some of the differences here. There's male sex hormones, female sex hormones, adrenal corticosteroids, mineral corticoids. I can never say that word the way I want to. Like aldosterone, and like glucocorticoids like cortisol. So we'll take a look at those. So here's some steroid hormones. These are some of the sex hormones, I believe. We've got testosterone here. We've got estradiol, progesterone, and then we've got a synthetic one, just kind of comparison purposes. So you can see here they all have the steroid backbone. Again, differences in the specific functional groups that we're going to see on these. Differences in their modifications. Similarities in the synthetic versions, right? And that makes sense. If it's meant to do something similar to the molecule that it's copying, well, it should have similar shape to that. So there's some adrenal corticosteroids. These are produced by the adrenal glands. These are located on top of the kidneys. This includes aldosterone. Do I have one here? Aldosterone. This regulates electrolytes and water balance. So that's what these adrenal corticosteroids do. Is they're electrolyte type balance, as well as they can regulate glucose levels. And so cortisone, no, cortisol, no, cortisone and cortisol. Both of these differences, right there. There's subtle differences here in this group right here. Both of these can actually increase blood glucose level and stimulate the synthesis of glycogen in the liver. So they're involved in maintaining a balance of stored sugar and usable sugar and everything else, electrolytes, all those things. So chemical messengers, piloting our bodies, but they're steroid based, again, from their structures. Identify each of these as a fatty acid, a steroid, or a triacylglycerol. So this should be easy. Pause it, give it a shot. Yeah, we definitely go into a lot more detail in my regular biochem class. We go through all the different hormones, and we talk about how they work and what they do. And I forget that this class is more of a topical class. All right. Last topic, really. This is our cell membranes. So here we want to talk about how substances are transported across the cell membrane. There's going to be a few different ways. We'll talk about diffusion, facilitated transport, and active transport. And the goal here, describe composition and function of this lipid bilayer. So, cell membranes, semi-permeable. That means they have to have the ability for things to pass through them, nutrients and waste and whatnot. They have to have cellular contents that are separated from the external environment. They basically protect everything inside the cell. There's a lipid bilayer that has two rows of our phospholipids that we've been talking about, the polar and nonpolar lipids. And they have an inner portion made up of the nonpolar tails and the polar heads on the outer and inner surfaces. So we're going to take a look at that. I already showed it to you, sort of, kind of was drawing it. So polar groups on the outside here, nonpolar tails, all oriented on the inside, and then more polar groups on the outside here. So one of these is the outside environment, which is like liquid, water, I should say, aqueous. And then on the inside of the cell, it's also aqueous. So that's why we've got that double layer. It's not enough just to have a single layer. It would be fine if we had nothing but nonpolar things inside it. And if you go back and take a look at the lipoproteins, that's exactly what they are. They're single layer because they only want to keep nonpolar things inside. We want to have polar things inside, so we have to have this bilayer. So, bilayer. Now, inside this bilayer is embedded lots of different things. We've got proteins and carbohydrates and cholesterol molecules. We've got pieces of protein that span the inside to the outside. We've got signaling mechanisms and receptors and all kinds of stuff happening. We're not going to go into all that detail, but it's very dynamic. It's very full of different things. They call it a mosaic because of how many different sort of parts go into it, and fluid because everything can shift and move around. Like this can move through here and pair up with this guy, and all of these things are free to move past each other within the bilayer. So I encourage you to watch a video on YouTube about lipid bilayers, an animated video, so that you could see kind of how they move. And nowadays they have some really nice computer-generated ones that are pretty true to life. So the bilayer also has proteins and carbohydrates on the surface that are good for communicating, and again, I said receptors for those hormones and neurotransmitters to actually bind to so that we can have cells communicating to each other. Now, the way that materials can get inside and outside has to do with what kind of transport mechanism we're talking about. So there's a few different transport mechanisms. Now, the first is called diffusion. Diffusion is a passive transport, so it requires no energy. No energy. It also requires no special transporter. So there are some things that can move in and out of the cell, and they only flow in one direction. They flow from high to low concentration. So that could be high on the outside, low on the inside, so it would flow in. But it could also be the opposite. It could be high inside, low outside, so it would flow out. But this would be passive transport, and it's just diffusion. Diffusion across the membrane from high concentration to low. Things like oxygen, carbon dioxide, water, all of these things can typically move between the bilayer. So then we have facilitated transport. So facilitated transport is going to be a situation where we have to have some kind of protein channel or membrane protein or something that's going to act as the gateway. And this also uses the concentration gradient. It'll go from high to low, same here, high to low concentration. So it just uses that normal mechanism to move across the cell. It's just as fast as diffusion, but it uses a protein channel. These protein channels are really good at being selective. So, for example, a potassium ion is a lot bigger than a sodium ion. So, if there's a particular channel that potassium or sodium is supposed to go through, it would prevent a potassium from going through there just by size. There would be other mechanisms like charge and that sort of stuff that could selectively keep one ion out, but usually size is good enough. And so again, this has to be a high to low um, direction, and facilitated here means that there's a protein, but again, no energy is required. Lastly, we have active transport. This one requires energy because one of the main points of this is that it moves from low to high. And you would say, well, why would we need to do that? Well, I mean, we, we we want to be able to move things in any direction that we want, right? And so nature takes advantage of the fact that high to low is this sort of spontaneous thing. But it makes sense that we don't always want to go with whatever nature's wanting us to do, right? If that was the case, we'd always walk downhill because it's hard to walk uphill. So to reverse this and to get something to go from low energy or high concentration, sorry, low concentration to a high concentration to kind of fight against that that concentration gradient, you do have to put in energy. These also typically require that there's a particular protein or a channel, something that's going to allow the molecule that we're moving to actually pass through the membrane, but it also requires usually ATP or some sort of energy. We'll call it ATP for now. You can just call it energy if you'd like. We'll go over the different energy currencies later. But for now, the three different transports are no energy diffusion, no energy facilitated transport, and energy requiring active transport. Um, Last learning check, and then I guess we're done for this chapter. So, which one of these moves particles across the cell membrane from high to low concentration? Um, yeah, of course, it doesn't exactly say if there's a protein involved. I guess since it doesn't say there's a protein, that sort of eliminates one of your answers. Yeah, I guess. So, if it said using a protein or an ion channel or some sort of, you know, passageway, we would call it facilitated diffusion. All right, so that would be your concept map. So again, the goal here is to be able to look at a lipid and classify it, know what kind of category it would fall into, know what components make up that lipid if it happens to be one that's hydrolyzable. If it's a steroid nucleus, we don't have to hydrolyze those. If your book goes into any information about the transport of cholesterol, I like to ask questions about that, so I would read over that if I were you. And definitely practice your hydrolysis, your hydrogenation, and your saponification of triglycerides. That being said, we don't ever really draw hydrolysis or um, saponification of phospholipids, so you should be good there. Though you could try it; it wouldn't hurt you. You would just get sphingosine instead of glycerol. But just focus on the triglycerides with your reactions. All right, everybody. See you in the next one. Take care.